So I'm really excited to introduce the speakers in today's session, which will focus on networked biology. And I have their names and their talk titles here for you to review, and I just want to say a couple of words about this session and about the general themes, and then I'm going to kind of pose the questions that I hope to address during the panel session of this, so that maybe if you guys have expertise in any of these or thoughts that could help us along, you can come to the podium or you can come to the microphones and help us all out. So the themes that we're going to be touching upon are really networked biology in social media, in the online social media, as in particular for patients. How do patients interact with each other? How can patients accelerate the pace of research? How can patients help each other by sharing data? How can data itself be better shared? How can genomic data be shared globally? And how can we all participate in order to generate more crowdsourced data? And so with that, I'm going to go ahead and start with my own talk, which is partnering with patients to accelerate the pace of genomics research. So I'm an associate director at the Broad Institute, where Sonia and Eric are also working, and I'm at the interface between patient advocacy and genomics research. And I'm not going to go into my background in any great detail, but I am a postdoctor. I've done a postdoc in cancer immunology. I'm a biochemist by training. I'm a patient. I'm surviving a very rare and aggressive cancer called Angiosarcoma, and I'm a patient advocate and I've stood up a foundation to try to bring awareness and funding to that. So if you want to talk funding at some point, we can, we can cross paths there too. And I was about ready to go down the traditional pathway of any academic scientist and take on a role as a PI. I had an MD Anderson faculty position waiting, and I just took a step back and thought, I'm not going to have the impact that I need on this exceedingly rare cancer. There's no way I can do this and actually be solvent and funded. And so I looked for any opportunity where I could really be at that interface between advocacy and any big resource, genomics, cancer immunology. I didn't care. I just wanted to be at something that could be bigger than one lab. And I landed at the Broad, and man, is it big. They have some really big, really exciting science there. And when I got there, I was there largely to answer this question. So my first slide is, can we engage patients to be the next revolution in genomics? And does that even make sense? Do patients want to be involved in research? And if they do, what are they going to even bring to the table? I'm a trained scientist, 15 years at the bench. The thought of working directly with patients at the time before I became ill was so far off the radar that I don't know that I would have even considered it at the time. And then, how? If we decide that the answer is yes to the first two, then how do we actually go about including patients along the entire process? We can't just build something and then say, come on board, you have to have them build it with you. So I came to the Broad to work with patients to accelerate genomics research. And the big question was, can the Broad work directly with patients to obtain unused tumor samples? Like, that's the ultimate goal. Can we obtain the unused tumor samples to do great genomic research? But to do that, really, there was a lot of other things that we needed to do and that we needed to do very, very well. We needed to engage patients in a meaningful way in order for them to be the biggest stakeholders in this entire process. So it turns out that only about 5% of cancer patients are actually enrolled in clinical trials. And so that means that only about 5% of people with cancer have their tissues processed in a way that's meaningful for research. 85% of US cancer patients are treated in the community setting. So they have a resection done, and then that gets stored away as an FFPE block somewhere on a pathologist shelf, and it never gets looked at again. So the thought was that we could work with organized patient groups, and we could transform the landscape of genomics for both rare and common cancers. And we live in the right time to do this. We couldn't do this 10 years ago. It's really only within the last couple of years where we've all become so networked. We've all become integrated with each other. As Eric and Sonia had mentioned, the internet is a wonderful thing. And when you have a rare disorder, the first thing you do is you find other people with that same disorder. If you have a common disease, you find the same people that have your particular flavor of that disease, and you become networked and you learn from them. So we are starting our studies at the Broad in metastatic breast cancer, and we have specific scientific questions that we want to answer while working with the patients. They're very ambitious questions. What are all the genes and mutations that can lead to metastatic breast cancer? What explains why some patients have extraordinary responses to a particular therapy? This right here is very important. If we can learn this, then we can apply it to people who may not know that they have the genetic underpinnings to have an exceptional response to a specific therapeutic intervention. What explains why some tumors never respond to a particular treatment? Why do people have acquired resistance? What genetic changes explain why some tumors initially respond to therapy and then recur and metastasize? What are all the genes that could lead to developing metastatic breast cancer at a young age? And ultimately, how can we better develop treatments for metastatic breast cancer? So we embarked about a year ago on generating this project where we have IRB approval and we've launched to collect patients' online consents. This allows us to go outside the borders of any institution and go directly to where patients are. And what we're doing is we're collecting information through surveys. We're collecting the consent to obtain people's medical histories. So we collect full medical record data sets. We get permission to obtain their tumor tissue. And then we perform, and we also send a saliva kit so that we can perform whole exome sequencing and generate data for tumor normal matched and also do transcriptome analysis. And then we're going to generate a clinically annotated data set and just share it with everybody. So this is what the site looks like. And we built this site along with every word and aspect of this in lockstep with the metastatic breast cancer community. And this picture up here is it's stock photo, but it was the most challenging thing to find. Just this picture alone, something that resonated with the patients. I had about 10 different iterations that I had gone through with patients, and they were super offensive for reasons that would have never occurred to me. You have, two white people on there. Well, what about, people of color? You have two old people. Well, what about all the young people that get this? You have a panel of patients that get the disease. Well, what happens when they die? And so everything that that we came up with and thought and floated by the patients was, balked at for some reason. And when I got this, the worst I got was, eh. And so we went with it. And so one of the criteria that we kind of set for ourselves was to make entry into this as low, the barriers to join and participate as low as possible. As a cancer patient myself, I have participated in research studies, lots and lots of them. I've been sent saliva kits, and I've also had saliva kits sit on the shelf of my office for 6 months without paying any attention. And so I knew that we can't have password protected anything because people don't want to take the time to do that. They want to just something easy, something quick, but still super highly engaging so that they know that they're really driving the research. We launched in October, and so far we've had over 1,800 people from all 50 states represent, all with metastatic breast cancer. And the patient-reported data that they're generating and giving us is allowing us to answer or even ask questions that we didn't even know was possible when we started this study. And I'll get to what those are in just a second. But I want to point out that what the enrollment has looked like. Okay, so we've had a bunch of different ways that we've tried to get the word out about the study. We leveraged social media, so mostly Facebook and Twitter, where we just started really engaging with different communities that were involved in metastatic breast cancer research. And what we found was that we could have articles in the Wall Street Journal written about this, and it would give us a little bump in enrollment. So you can see that. Well, there's a Boston Globe article right here in the middle of our launch, and it gave us a little bump. And we had a Wall Street Journal article, and it gave us a little bump. But what really sets off the enrollment is when popular people in social media post about this. We had our biggest day of enrollment was right here, where we enrolled over 300 people from one Facebook post, from one patient who had passed away a month before, and somebody posted on her behalf saying that it was important to her. And so we had people go to bat like never before, saying that if it's important to this person, then it's important to us. As a result of the surveys that we ask patients, the answers that they've provided us have allowed us to identify cohorts right up front. It's one thing to have your hands on people's medical records, which are this big, but it's another thing to have kind of a very nice, neat little collation of their entire experience with cancer up front so that you can see immediately, does it look like these people have had extraordinary responses to therapy? Are these people young? Even that alone is a good differentiator in order to get the cohorts that we need in order to do these meaningful studies. And as you can see, what we wanted to find, one of the very first things that I had mentioned is that we wanted to identify people who had extraordinary responses to therapy. And it turns out that we found many, 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 way more than we thought. You know, when we had no idea, this whole thing is an experiment. We had no idea what to expect when we launched. We thought that it might take a year, and it might take 5,000 patients in order to find 100 needles in a haystack of people that may have had exceptional responses to therapies. But within the first couple of months, we had hundreds of people who had extraordinary responses to therapy. They were either on a specific therapy way longer than would be expected before they developed resistance, or they had had complete shrinkage of their tumors on a certain therapy that we had no idea. And if you go to Dana-Farber or MD Anderson or Sloan Kettering, you're not going to find 100 people who have had extraordinary responses to Xeloda. It's not going to happen. But we have them here. And so now we can get their tissue and we can look to see why. Why are they different? And is there an underlying genetic reason that they're different? And can we then look prospectively in other patients in order to identify them de novo? What is really at the crux of this project and the way that the Broad is really thinking of their current and upcoming projects is the patient engagement aspect of this. So I said in the first start of this that the point of this was to get unused tumor samples. But that sounds very dry. When you really think about what the point of this is to see, can patients actually accelerate the pace of research? Do they want to participate in research? And we found overwhelmingly that the answer is yes. And I'm not going to read every single one of these quotes from patients that are participating in it. I'll just read the first one from this woman, Allison. This really encompasses why people are participating in this. We asked her what the project meant to her, and she said, I want to live and watch my children grow up, but if I can't, then I want to leave a legacy and a cure. And that is overwhelmingly what we get from the participants. They know that this research is not necessarily going to come in time to save their lives, but they want to know that what they're going through is not in vain. They want to contribute so that other people coming down the pike have a huge wealth and a bunch of resources available to them. So we have gone out of our way to find ways to meet a lot of these patients in real life. So every time we go to conferences, we have meetups, and there's inevitably 10, 15 patients that'll be there. We invite them to the Broad. We try to get them to interact with the scientists so that there's not just data that are nameless and faceless. I think you may have noticed that on all of my slides, there's always a picture of a patient that's participating because they're not data points, they're patients, they're real people. As for the next steps for this particular project, we're scaling up. We have now sent consent links to everybody who has registered for the project, and we are in the process now of sending out saliva kits to everybody. So by this time next week, 1,000 saliva kits will have been mailed out. We just started mailing the saliva kits out last month and have received almost 300 back, which is great. We have already done medical record requests and tumor sample requests from a number of the people that were in the initial cohorts and received them back, and have our first data back from the genomic analysis, and our bioinformaticians are actually analyzing that right now, which is very exciting. We're moving into an era where it looks like we'll be able to do whole exome sequencing from blood biopsies, and so we're piloting that with a cohort of about 300 of the patients, and we anticipate being able to do that this summer in a pilot phase, and then launching that in the fall. And then we're just going to generate the data, and at every 6 months, we're going to do a data dump and make that available to everybody. And what we've learned is that when we go back and ask that first question, do patients want to be engaged? They do, very much so. They have the desire to be involved in research. They form organic communities, and they have extensive wealths of information. And they have the ability to crowdsource their own tumors. And if you partner that with a big institution like the Broad, you could do large-scale genomics, generate these giant shared resources that can be shared by the entire biomedical community, and you can have high impact using unconventional methods. So I'm just going to talk very quickly about the next project that we're rolling out. You know, we thought that maybe we would go into the space of rare cancers, because just like you can't find 100 people that are going to have an exceptional response to Xeloda, you're never going to find 100 people that have a tumor that only affects 300 people a year worldwide. It's not going to happen. Even at the major institutions, you're only going to have maybe 9 or 10 a year that walk in there. So if you think about the space of rare cancers, which really are about almost half of cancers, depending on how you define it, that's half of cancers that are largely going under the radar in the research world. But this, going directly to patients and having a very low barrier for them to participate, is an ideal way to get them enrolled and get those tumors sequenced. You know, there's small numbers of patients, so you could effectively sequence an entire living cohort of people. And the impact is very large. And the way that I like to sell this to scientists is, if you want a paper, pick any rare disease and study it, because you're going to get it. It's going to be published because nobody knows anything about it. I said I put a patient on every single slide. This is actually me 6 years ago after I was diagnosed with a very rare cancer called Angiosarcoma. As I had mentioned, this is a very rapidly proliferating, extensively infiltrating malignant endothelial cell cancer. It has the poorest prognosis among all tissue sarcomas. It's exceedingly rare. I had angiosarcoma of the breast. So that's normal breast tissue, and this is what breast angiosarcoma looks like. This may be my tissue, I'm not quite sure, but it was provided by my pathologist. And you can see on the Kaplan-Meier curve that it's a very steep, it's a very steep decline. I always usually present this with like a little happy face right there, because I'm just like watching myself kind of trek down the Kaplan-Meier, and I'm still here, which is always a good thing. And we do know some things, because my organization has funded research in this area for the past 6 years. It's sporadic mostly. We don't know what the genomic causes are. It hits everybody, kids, elderly, every race, creed, and color. There are certain demographics that are at higher risk. Elderly Caucasian men usually get it in the head and neck. People around my age get primary of the breast. Sometimes people that have things left in them during surgery will get angiosarcomas that rapidly grow around them. One alarming alarming demographic that's increasing are people that have conservation therapy after breast cancer. They'll notice a little bruise about anywhere from 7 to 11 years after, and that's an insidious hint that angiosarcoma is arising. And it may go years they may go years thinking that they've just bruised themselves, and by the time they go to the clinic, they're metastatic. The published genomic drivers of this are right here. I can show you right here. So we know that there are some players that have been published upon. But what I found super interesting is that because of a Facebook group that I'm part of, we started sharing our own Foundation 1 reports. And the patients' genomic information did not overlap with what was published in the literature. So they were turning up mutations that the literature and the scant data that we had did not turn up. So that means we have far way to go before we exhaust the list of drivers of this disease. So as I said, it's an exceedingly rare cancer, but I am part of this Facebook group. And when we decided to launch this study as proof of concept, I said to my colleagues at the Broad, I said, I could get you guys 100 people in one day to be part of this. And they said, really? And so I just posted this. I said, by a show of likes, if you're living with this disease, would you consent to let us use your tissue? And within an hour, I had 92 people, 1 hour. So that is just proof of concept that if you have organized patients, you can leverage them to really actually do very, very rapid research that you could never do in any other context. So we're building out the Angiosarcoma Project at the Broad. We are going to launch it this summer. I'm really excited. We're just putting on the final touches to building out the database that's going to hold the data. This is Jim. He's a 32-year survivor of this disease and a beacon of hope to all of us. And I guess I'll just end with, where there are patients, there can be projects, okay? And so it's good to keep our minds open as scientists that, there's all kinds of space to engage patients along the way in space of genomics, rare cancers, outliers and common cancers, find interesting phenotypes. Other stuff that you can do is you can create models, cell lines that don't exist yet. And at the end of the day, the patients are the biggest stakeholders in our collective efforts to thwart this disease. Lots of people to thank. So Nick Wagley is the PI of the Metastatic Breast Cancer Project, and of course, always the patients.